Hello everyone, my name is Brenna and today we will be discussing the viral evolution of HIV. HIV is a positive sense single-strand RNA virus. It is closely related to SIV, which is simian immunodeficiency virus. SIV is found naturally in non-human primates. Specifically, SIV-CpZ and SIV-GOR are thought to have been the original virus that HIV evolved from. CpZ indicates that that strain was from the West Central African chimpanzee, while GOR indicates that strain was from the Western lowland gorilla. And the jump from non-human primates to humans describes a phenomenon called cross-species transmission, which is the transmission of a pathogen between hosts of a different species. Additionally, we will be discussing single nucleotide polymorphisms, or SNP. This is a loci with alleles that differ at one base, and it's more commonly known to us as point mutations. Toll-like receptor 4, or TLR4, is a type of pattern recognition receptor that serves to prevent infection by initiating an immune response. So this shows the importance of TLR4. HIV enters the cell via CC chemokine receptor 5 or CXC chemokine receptor 4. TLR4 then recognizes the virus and causes a downstream immune response. And so in that way, the stimulation of TLR4 can help to inhibit HIV-1. However, if there is an SNP or mutation of TLR4, then the risk of HIV increases as the body is not able to initiate this immune response. CD4 T cells are important to that immune response. These are single-chain molecules made of 4 immunoglobulin domains. CD4 T cells bind to the beta-2 domain of the class 2 MHC molecule. MHC molecule is the major histocompatibility complex. And class 2 means that this is the type present on antigen-presenting cells. Humanized mice, or humice, contain a transplanted human immune system. So they inject these mice with human CD4 T cells, macrophages, and dendritic cells. The reason why those 3 cells are included is because there is research supporting that these are the cells HIV attacks. And that is why there can be such severe neurologic symptoms associated with HIV and the subsequent AIDS infection, because macrophages and dendritic cells are important to the functioning of your neurosystem. The framework of this research, our goal was to investigate viral evolution in terms of HIV specifically. And in doing so, the questions I hope to answer were: how did HIV evolve? Where did HIV evolve from? What caused the evolution? So could we identify any selective pressures? And additionally, what impact on the host does SIV have that allowed it to continue to live and become HIV? To investigate these topics, the focal paper I used was the SIV-CpZ cross-species transmission and viral evolution to HIV-1 in a humanized mouse model. To investigate this phenomenon, the researchers transfected human embryonic kidney cells with the SIV-CpZ EK505 plasmid. So that's just a specific strain of SIV-CpZ. They injected 200 microliters of the virus into the abdomen of 5 humanized mice, and they tracked the impact of the virus by doing blood draws to find the plasma viral load and the CD4 T cell count. Additionally, they tracked the genome with viral RNA extraction using reverse transcriptase PCR. And they did this at the 3, 11, 19, and 22 week points. And this was done with next generation sequencing, which is just using DNA or RNA to sequence the genome. So at each of these weak points, they compared the genome that they extracted to the genome of SIV-CPZ stock virus of the strain that was used. And to identify the variations, they needed to see a greater than 20 reads depth of coverage and greater than 20% frequency increase of that mutation relative to the stock virus. And this greater than 20% threshold just had to be met by the 22-week point. So it was after the completion of the entire experiment that they determined which variants were significant enough. So what they found in reference to the viral load was that after 7 days, the virus was detectable in the plasma of infected mice. So right here, we see the start of detection, and it increased over the period of 80 days post-inoculation, where it peaked. And after that peak, there was a gradual decline. The total increase of viral load was 2 orders of magnitude. In regards to the CD4 T cell count, there was a decline of CD4 T cells in the infected mice over time post-infection. So we see here that right at the point of infection, there was a quick drop-off of CD4 T cells in the infected mice. There was also actually a drop of CD4 cells noted after about 10 days in the uninfected mice. And that allows us to see a really important trend, which is that the uninfected mice were able to replenish their CD4 T cell count. And they actually finished at the completion of the experiment with a greater CD4 count than they started with. However, the infected mice, after this initial drop-off at the time of infection, were never able to replenish their CD4 T cell count stores. And they finished with a lower amount of CD4 T cells than they started with. In terms of the genome mapping, there were numerous synonymous and non-synonymous SNPs in the genomes of measured from the infected mice. So a synonymous SNP is the same as a silent mutation. So this is where the amino acid does not change. Whereas the non-synonymous SNPs would change the amino acid. So there were a total of 18 genes that met this greater than 20% increase threshold at the 22-week post-inoculation time point. The gene with the highest endpoint frequency was ENV at codon 403. The amino acid change was G to E, which is glycine to glutamic acid. So what this tells us about the transmission of SIV-CPZ into humanized mice is that SIV-CPZ was able to live in this environment despite the presence of the humanized immune system. And that's evidenced by the measurable viral load and the increase of the viral load for the first 80 days. So SIV is able to live in the system. It's able to reproduce and actually grow in number. The way that SIV-CPZ impacts its host is the reason why it was able to live for the duration of the experiment. So whenever SIV-CPZ was introduced, the CD4 T cell count quickly declined, and then it was not able to recover to its original values. And this relates to the immunosuppression that we see in humans with HIV. And the presence of the SNPs that were found shows how SIV-CPZ adapts to humans and actually changes the genome. And other research has shown that the frequency and location of these mutations can actually indicate the severity of HIV complications, again, specifically in terms of neurological disease with the CD4 T cells, macrophages, and dendritic cells. So essentially what SIV-CPZ does is it inhibits the immune system of the host, which allows it to mutate and just thrive in the body's system. So regarding the research questions that I started off with, I was wondering, how did HIV evolve? And we saw with the focal study that this was cross-species transmission from a non-human primate, which naturally hosts SIV, and specifically SIV-CPZ. The way that SIV was able to enter the human body system and evolve into HIV was because it impacted the host by decreasing the immune response. And the way it does this is with SNP induction or mutations and the decline in CD4 T cell count. Other research has also shown that mutations in TLR4 actually leads to an increase in viral load. So recall at the beginning of this presentation, we discussed how TLR4 is one of the molecules that notices HIV-1 infection. But mutations in TLR4, which can be introduced by the HIV virus itself, can actually prevent the recognition of more viral molecules, which will lead to an increase in virulence. However, we still have this unanswered question of why did HIV evolve from the original host? What was the selective pressure? There have been theories that this occurred whenever humans started interacting with non-human primates more, possibly through field research, that SIV, which is known to mutate a lot, was just able to mutate to further infect HIV or human hosts. So there is further ongoing research using various plasmid strains, more than what we examined in this current focal paper. And again, we still don't know these selective pressures, so in the future that will be a good main point of research. So thank you for listening to my presentation. Here are the research I cited, and I look forward to answering your questions.